Hey everybody. Okay, so this is part one of the chapter 10 lecture. Um, Chapter 10, or the info for this week, will be split up into three sections. So this is part one, and part two will talk about independent samples t-tests, and part three is the practice for the math for independent samples t-test. All right, so let's get started. So chapter 10 is just a quick introduction into simple experiments. Simple experiments being one independent variable and one dependent variable. So super simple. As we get into chapter 12, 13, we'll talk about studies with more than one independent variable, and it gets a little more complicated, but not too complicated, so don't get scared. Okay. So for this week, we're going to cover some of chapter 10. The second half of chapter 10 will be covered next week, and that's the within groups design t-tests, and then we'll interrogate our causal claims with our four validities that we talked about last week. So this week, we're going to talk about two examples of simple experiments, and we're going to use these kind of like to as examples for what we're going to talk about. We'll talk about what are the experimental variables, specifically our independent variables, dependent variables, and control variables. We'll talk about why experiments are important for supporting causal claims, specifically more so than association claims or frequency claims. And then part two and part three, we'll talk about t-tests for independent groups design, which is one of the two types of simple experimental designs. So first thing first, two examples of simple experiments. So the two examples that we're going to talk about are in your textbook. Example one is our taking notes study, and example two is eating pasta study, which personally I would love to be part of because pasta is life, or carbs are life, but no one invited me. Okay, so example one is our taking notes study. So what this study did was, let me pull up my little laser pointer. What this study did is they split participants up into two groups where they either had these participants take notes using a laptop or take notes pen and paper longhand, right? Old school. And then they gave them a series of tests that had factual information and conceptual information in the test. And then their dependent variable, the thing that they measured, was the score on their essay questions, which were standardized. And so what they found was that people that took their hand notes versus laptop did equally well on factual information. So if you're just learning facts, it doesn't matter if you're taking notes by laptop or old school on pen and paper. However, for conceptual information, which hint, is pretty much 90% of the stuff that you learn in college, right? You learn a bunch of facts, but you also learn about concepts and theories and how to apply these facts. Anyone that took notes longhand did significantly better. They scored significantly better than those that took their notes by laptop. What this should suggest is that taking notes longhand on pen and paper, even though it takes forever and it's a lot of effort and your arm starts to hurt or your wrist starts to hurt, it actually allows for better retention, which is why you do better on these essay questions. Our second example is our eating pasta study. And this study is amazing. As I mentioned, I would love to be part of anything that has to do with eating pasta. And what they did is they wanted to see if the size of your bowl or the size of your plate impacts how much food you consume. Okay. So what they did is they had two groups, and one group got a medium bowl, and the other group got a large bowl. So those are one independent variable, bowl size, two levels to this independent variable, medium and large. And then they just measured, they took them to a buffet. Oh, tell me this study isn't amazing. They took them to a buffet and gave them either their medium bowl or large bowl, and then just measured by grams how much pasta they served themselves. And then they also measured separately how much calories was consumed based on calories per gram. Okay. And what they found is that individuals that had a medium bowl took significantly less pasta than those that had a large bowl. And this reflected in those that had a medium bowl ate significantly fewer calories than those that had a large bowl. Okay. So those are two examples of our simple experiments. As we'll talk about the next 3 bullet points, we'll bring up these experiments kind of just to have as examples. Because if I just talk to you about what is a variable without examples, it's even more boring than this lecture already is. It's okay. I know. I'm not offended. All right. So experimental variables. We have independent variable, dependent variable, and then our control variables. So an experiment is formally defined as where you have a study where at least one variable is manipulated and one variable is measured. So if you remember a correlation or an association claim, right, we run correlations, you have 2 measured variables. You're not actually manipulating anything. And so this is, in an experiment, you have to have at least one variable that is manipulated, that you're separating people by into groups. Now, you may notice in some of our examples that will follow, not yet, that we might have sometimes an independent variable, the thing that you typically separate people on into groups, that is not exactly manipulated. It's a measured variable that we decided to split people on. So for example, imagine I wanted to examine sleep hours, difference by groups of age groups. And so let's say I have teenagers, young adults, older adults. Age is a measured variable, right? It's not a I can't manipulate your age. I can't be like, oh, you're 17, but I really need you to be 35 for this study. So could you just go into the 35 age group? That can't happen, right? It's a measured variable. It's a variable that I measure, and then I see what your age is. And so if I wanted to see differences in sleep across age groups, both of those variables are technically measured. But if I take people's measured variable of age and I split them upon this construct about this variable, then it becomes what we call a quasi-experiment, where the independent variable is not able to be manipulated, but we can split people up on these variables, right? I can say teenagers is 12 to 19, adults is 20 to 40, older adults is 41 to 65, right? I can split people up on this manipulate and create almost like a pseudo manipulated variable. And so a quasi-experiment is still experimental because we're splitting people up into groups, but it's when you can't technically have true random assignment where you randomly place people into 3 groups, right? I can't randomly put a person from the population and say, you're going to go into the teenage group because they might be 60, okay? So just to elaborate, a manipulated variable is typically the thing that a researcher assigns you to that group, and it's something that they are manipulating, something that they are adjusting. So in the pasta study, for example, it's the manipulated variable is the bowl size, right? That's the independent variable. The independent variable is what you're manipulating, and it's one independent variable, bowl size, and it has 2 levels, medium and large. And so it's the thing that the researcher is wanting to see if it has an impact. In the pasta study, what they wanted to see was whether the size of the bowl impacts how much you ate. And so their independent variable is that bowl size. And then we have a measured variable. Measured variables are also typically dependent variables. This is the thing that you're wanting to see if it has an impact on. This is your outcome. So it's what you record, it's what you measure to see if the independent variable has an impact on. So in the pasta study, the measured variable, the dependent variable, is how much pasta was eaten, or the number of calories consumed, right? It's the outcome. It's the thing at the end that you're seeing if the independent variable has an impact. You're seeing if bowl size had an impact on pasta eaten, okay? So that's our measured variable or dependent variable. We also have control variables. Control variables are not things that you necessarily measure or manipulate, but rather, if you think ahead of time of a variable that could potentially impact your findings, could potentially impact the effect on your dependent variable, but is not your independent variable. So think about in the pasta study. So we want to see if bowl size impacts how much pasta you eat. What else other than bowl size could impact how much pasta you eat? So just think for a second. Could it be maybe the type of pasta? So maybe penne is better than fettuccine. Or maybe some people got macaroni, those tiny little U-shape, smiley face shapes, and some people got spaghetti. And maybe there's a preference for spaghetti over macaroni, right? What about the type of sauce? So maybe some people had marinara, some people had alla vodka sauce, some people had cream-based Alfredo sauce. So you want your control variables ahead of time when you're designing your study, besides making sure that you vary bowl size and you're measuring your pasta eaten as your dependent variable, you also want to make sure that you're using the same pasta, you're using the same sauce, you're presenting the food at the same time, or whatever else you can think of that might be impacting your study. And these become your control variables. These are the variables that you make sure are the same between your two groups, between your levels of your independent variables. All right, so that's it for our experimental variables. Now we're going to talk about why experiments support causal claims. So before we go into that, I really want to say that causal claims are really bold claims. Like, you're saying that something that you're manipulating actually has an impact or changes something else, right? It's a really bold claim to say that this has an impact on something else, because association claims are really easy. You're just seeing if there's a relationship between them, but you're not saying that one causes the other. You're just saying, hey, this might increase the chance of this happening, right? And so causal claims are really, really bold, and therefore we have to make sure that studies that use and make these bold claims are actually able to test causal claims. And that's why experimental design is so important and so complex. Not to scare you, just, we'll get it down, but it's important to really understand the ins and outs of experimental design. So experimental design is the only one that can determine cause and effect if it's designed well. So when we think about association claims, so go back to 3510 for a second, association claims. Often we talk about an association, a relationship between studying and exam grades. And the relationship is really strong. So it's usually a positive 0.8, where the more you study, the higher your grades, or the higher your grades, the more you study. Right? But we can't tell there the cause and effect. Why? Because the only thing that we have out of the requirements for a causal claim is covariation. So a causal claim has these three requirements. It has covariation, which is essentially a relationship. What it means is that you should see a difference in your dependent variable based on your independent variable. Okay? Causal claim also shows temporal precedence, which essentially means that we know that X came before Y. We know that the independent variable was presented before Y. So in a correlation, we don't have directionality. We don't know if studying causes or came before taking the exam, or maybe taking the exam came before you actually sat down to study or to review. We don't know if in another correlation, for example, there's a relationship between getting older and number of sex partners. So the older you get, the more sex partners you tend to have, and the relationship is pretty strong. It's like 0.52. And so we could say, oh, well, the older you get, that happens before increase in sex partners, but we wouldn't say the other way around. We don't have the directionality, and so we can't, but it would be weird to say more sex partners makes you get older, right? That's very strange. And so a correlation just states the relationship. It can't tell you about the directionality. We don't know which came first. You're just measuring two variables. You don't know which happened first. And so an experimental causal claim, experiments support a causal claim because it establishes for sure we manipulated X, we manipulated our independent variable, and then afterwards we manipulated, we measured our dependent variable. And then the third thing is that it has internal validity, which is essentially another way of saying is that we rule out other variables, third variables, what we call confounds, or the third variable, or extraneous variables. So many words. So we can control for variables that we predict ahead of time that would impact our results. And so we want to make sure that our internal validity is high. But let's talk about each of these. So one, experiments establish covariance. Covariance being that we know that the dependent variable, the thing that you're measuring, the outcome, let's say how much pasta you ate, varies depending on your levels of your independent variable or your comparison groups. Medium bowl, large bowl. So you may remember from 3510, we talked about why we don't want to think about intuition. We don't want to just make the hunch like, oh, of course the size of the bowl impacts how much pasta I eat. Every time I take a large bowl, I eat more pasta. Well, how do you know that, right? We can't just use our intuition. We have to have a comparison group. And so comparison groups are inherent in experiments because even if you only have one independent variable, you have to have at least two levels. And so that automatically gives you at least one comparison group. So you're comparing medium bowl to large bowl, or you're comparing longhand notes from laptop notes. So you have an inherent comparison group to examine. And there's different types of comparison groups. We have just comparison groups, which are different conditions. We have control groups. Control groups are where you have a group that doesn't get a treatment at all. And so this is super common in designs where you put somebody into some sort of situation, and then the other person, you just give them your measure. So let's say I want to understand the effect of watching happy movies on depression. So I can take two groups, and one group I make them watch happy movies. And in the other group, I don't make them watch any movies at all. They just take a depression questionnaire. And so one group is the comparison group, or the treatment group, or the condition, the experimental condition. And then the other is the control group, where they don't get any level of the independent variable. They get nothing. They just get the dependent variable. Then we have a treatment group. Treatment groups are very common in treatments. So for example, if I wanted to understand, again, the effect of depression, movies on depression, so the treatment group might be one group gets a happy movie and another gets a sad movie, right? Or a horror film or something. And then I give them the same dependent variable, the same depression questionnaire at the end. And then we also have our placebo groups. Placebo groups are very common in medical studies, or when we want to see the effectiveness of a drug. So a placebo control group is essentially where you give somebody something, but it is inert. It has no effect. So for drugs, for example, let's say I wanted to see the effect of a new drug on depression, just sticking with the depression example. And so I give one group 10 milligrams of my new drug, and I give the other group a pill that looks just like the 10 milligram pill, but it has nothing. It's just sugar pill. And this is a placebo. And I give them the same depression questionnaire. And if you find that those that took the actual 10 milligrams of the medication have lower depression than the placebo, that means that the medication works. But if you can also imagine that, let's say you take the 10 milligrams, one group takes the 10 milligrams of the medication and they get their depression score, and the depression score is the same as those that took the sugar pill. It's just a pill, but it has no effect. It has no medicine in it. If their scores are equal, that means that your medication was not effective, because that means that the placebo was just as good as taking your actual medication on depression. Now, two things I want to note, because often students get confused. If a study has no variance, it cannot be analyzed because you can't establish covariance. You can't establish differences between groups. If everybody in all groups scores the same, you have no study. You have a null effect, because what you're saying is that there's no differences between the groups. And the second thing I want to note is the word control variables is very different than a control group. A control group is one of your levels of your independent variable. A control variable are things that you think about ahead of time to make sure that you control for them: type of pasta, type of sauce, time of day that you're eating it, that to make sure that it has no impact on your study, it has no impact on your dependent variable. Okay, so a control group is a function of your independent variable. A control variable are variables that you want to make sure that are the same between your levels of your independent variable. Okay. Number two, and this is the easy part, temporal precedence. So experiments establish temporal precedence. We can see that the cause came before the effect. The differences in bowl size come before our measurement of how much you eat. And this is super simple to understand. I know for sure that the size of bowl came before your pasta size, your pasta consumption. And this is, by the way, just as a reminder, not something that we can establish always. Sometimes it's clear, but we can't always establish this clearly in an association claim, in a correlation, and definitely not in a frequency claim. And third, a well-designed study establishes internal validity, which means that it makes sure that the only thing that has an impact on your dependent variable is your independent variable. And so in order to do this, we have to make sure that we control for confounding variables, or confounds. So confounding variables are the things that we would think about ahead of time. These are the alternative explanations that threaten our internal validity, such as pasta quality, pasta sauce type, time of day, right? We thought about it ahead of time. What are these confounds? What are these potential alternative variables? And we use them as control variables ahead of time. Other confounds that we have to think about are selection effects and order effects. We're not going to talk about order effects very much, because they come up when we talk about a within subjects design. Right now, we're only going to talk about between subjects design, but we're just mentioning it. So a design confound, I want to make a very clear distinction, is only when the problem between the two groups is of systematic variability. So if we have random variability or unsystematic variability between the two groups, it's not a confound. We can't control for it. We can't think of it ahead of time. So an example of unsystematic variability is how much an individual likes pasta. Some people like pasta more so than others. Those that don't like pasta, who are you? But those that don't like pasta as much might still be in the group. But I can't control for that. That's not systematic variability. We should assume that the two groups that we have, our two samples, will vary on how much they love pasta, right? And that's okay. What we want to make sure is that we control for these confounds that are systematic variability, such as the time of day. We want to make sure we give them the bowls 20 minutes before lunchtime, right? So that we can kind of understand hunger level, right? People tend to be hungry around 11:30 noon, more so than they're hungry around 1:30, for example. We want to make sure the pasta type, because there are studies, awesome studies, that find that some pastas are more favored than others systematically. And so we're only worrying about systematic variability when we're talking about confounds. It's okay to have unsystematic variability. We can't make sure everything is the same. That would be impossible. That study would never exist. So we allow for unsystematic variability, just random variation between people, but we want to make sure we control for things that are systematically going to differ between groups. Okay, so one other confound that we worry about is selection effects. So selection effects is where participants in one level of your independent variable, let's say medium bowl, vary significantly from or systematically from another level of your independent variable. So for example, what if all the people in your large bowl group are men and all the people in your medium group bowl are women? Why might this be a problem? So think about, for example, if all of those people that had the large bowl were men and all of them that were with the medium bowl were women, why might that make our results not so reliable if we found a difference? Well, men, due to their on average larger body size, tend to eat more and need more calories in order to sustain their larger body, right? So men eat more calories, need more calories, and therefore consume more calories on average than women do on average, right? There are variability, but as a whole, there's lots of studies that show that men consume and require more caloric intake. And so if they all had the large bowl, we might find that they eat more, not because of the large bowl, but because they're all men, right? And so we want to make sure that our groups, the selection of who goes into our groups, is random, right? So one way to avoid selection effects is random assignment, where I just take the population in front of me, and when I sample from them, I randomly assign first person to group 1, first person to group 2, first person, second person to group, wow, I messed that up. First person to group 1, second person to group 2, third person to group 1, fourth person to group 2, right? I randomly divide them into my 2 groups so that I eliminate any systematic variability. I might have unsystematic random variability, but that's okay because I didn't intentionally assign different people to different groups. They were randomly assigned to the 2 groups. Now, one thing that you might have a problem is in small groups, you might still have some systematic variability. So if you're having a small group, even if you randomly assign, you might have 8 women in the medium bowl and 8 men in the large bowl. And so when you have small groups, one way to eliminate these selection effects is matching or matched groups, where essentially you make the 2 groups match on the variable that you think might be a problem. So if it's gender, it's sex, not gender or something else, but if it's biological sex, then you might take group 1 and make sure that they have equal number of males and females to group 2. And so that way you're making sure that the groups are matched on this variable that might be an issue. This is another way of controlling for that variable. Okay. The example in the textbook or in this slide as well is about GPA. So let's say you want to see if there's differences in intelligence between these 2 groups. But GPA is something that is very related, and we know influential on intelligence, on IQ score. And so you might want group 1 and group 2 to match on its variability of GPA. And so group 1, you'll take the 2 participants with the highest GPA and assign it to group 1, and then the 2 participants with the highest GPA and assign it to group 2. Then you're going to take the next 2 highest GPA participants and assign 2 to group 1 and 2 to group 2. And so you're making sure that the groups are the same on this variable that you think might be a confound. This is another way, again, of controlling and making sure that it's controlled between your groups. And then order effects. Again, we're not going to talk about order effects right now. We're going to talk about order effects next week when we talk about a within subjects design. We're only going to focus on between subjects design more or less for this lecture. So don't worry, we'll talk about order effects. Okay, so just as a review, why can experiments support these bold causal claims? Well, one, they allow us to see a change due to our independent variable. We call this covariance. Two, they ensure that the independent variable came before the dependent variable. We call this temporal precedence, again, not capable in a correlational or association claim. And they establish that the only thing that changed is the independent variable. And so if you see a difference in your dependent variable and the thing that you're measuring, then the only cause is your independent variable, because everything else was controlled for, or everything that we can control for, everything that's systematic is controlled for. And we call this internal validity. Okay. And last but not least, we're going to introduce our independent groups design, which is the basis for our stats for this week. So there are two types of designs when we talk about experimental design. There's more, but we're not going to talk about them today. We're going to talk about just the two basics. And they're either independent groups design or within groups design. We're going to focus on independent groups design, but we're going to introduce both. Independent groups design is also called between subjects or between groups. Within groups design is also called within subjects, repeated measures, and dependent groups. Okay, so an independent groups design is where you have at least two groups. You have one independent variable that has at least two levels, and you're dividing your participants into these groups. And there's two types of independent groups design: post-test only and pre-test post-test. A within groups design, also called within subjects, is where all your participants are exposed to every level of your independent variable. Okay, so this is very different. In independent groups design, you have two groups, and any one participant only is exposed to one level of your independent variable. And this is the main difference between them. In independent groups design or between subjects design, one participant is only ever exposed to one level of your independent variable. In a within groups design, any one participant is exposed to every level of your independent variable. Okay, so we're going to ignore the within groups design for right now. We're going to talk about that next week. And I'm going to go over these two designs, the post-test only and the pre-test post-test, right now. So independent groups design only. So we have a post-test only design and a pre-test post-test. So a post-test only design is the most basic, simple experiment for between subjects. Essentially, you have a group of people, and you randomly assign them to be part of either one level of your independent variable or another level of your independent variable. And then you give them the same dependent variable. So just as the example that we already are familiar with, let's say you have a group of students. You randomly assign eeny, meeny, miny, mo. You get split up into the two groups to either take notes with your laptop or take notes on pen and paper. And so the people in this group only ever take notes on laptop. They never also have to take notes on pen and paper. So they're either assigned to the laptop note group or the longhand note group. And then they're both given the same comprehension test. Okay? And so in a post-test design, you only are exposed to your one level of your independent variable, and you get the same dependent variable one time. In a pre-test post-test design, you get randomly assigned into groups again, but you get your dependent variable twice. You get your dependent variable first, Then you get one level of your independent variable, and then you get your dependent variable again. So for example, let's say I wanted to see whether mindfulness improves GRE scores on the verbal part of the GRE. The GRE is essentially the grad program version of SAT. What I'd like to call the GRE is the SAT on crack, because it's an awful test. Awful, by the way. Not trying to scare you if you ever have to take it, but it sucks. But anyway, so let's say your research question is mindfulness going to help you improve your scores on your verbal GREs? Okay, so I decide that that's one level of my independent variable. The other level will take some other class, a nutrition class. This could be a comparison group, but it could also be kind of like a placebo group, right? I'm making you take a class, but it has nothing to do with what I think might impact your scores on the verbal GRE, because nutrition shouldn't necessarily help you. So I'm making you take a class to make sure that it's not just the effect of taking a class that improves your score. And so what I do is I want to make sure that your verbal GREs in the beginning are whatever level they are, and I want to make sure I control for that after the class. So I give you the verbal GRE first. I give it to both groups. And then you either take part in the mindfulness class or the nutrition class. And then I give you the verbal GRE scores again. And what I'm looking for is the difference between the verbal GRE score here and the verbal GRE score here. Okay. And so in a pretest post-test design, you're still only being exposed to one level of the independent variable, but you get exposed to the dependent variable before and after that independent variable. Okay. And that's it for part 1. Part 2 of this lecture will discuss how to run an independent samples t-test, which is the t-test we use for between subjects design or an independent samples design. Part 3 of this lecture will go over the practice problems of an independent samples t-test. This will go over the math. This will be really important for you when you're doing your homework. So please make sure you listen to the voiceovers. The homework has 2 math problems, and you really want to feel comfortable doing that math because it will be on the exam. If you have any questions, please let me know. Oop.